Meiosis. DNA is replicated so each chromosome consists of 2 chromatids. A diploid cell is a cell that contains 2 sets of chromosomes.
Chromosomes shorten by supercoiling. Each consists of 2 chromatids when they become visible. This is due to DNA replication in the nucleus during interphase, giving each chromosome 2 sister chromatids. These chromatids are genetically identical. DNA is now replicated again between the first and second division of meiosis.
Breaks in DNA occur and non-sister chromatids invade a homologous sequence on a non-sister chromatid and bind to the region of the break. Once crossing over, non-sister chromatids adhere to the site where crossing over occurs. These connection points are called chiasmata. Evidence suggests that these connections are essential for a successful completion of meiosis.
Crossing over occurs at the same position on the 2 chromatids involved, which creates a mutual exchange of genes that promote genetic variation. In random orientation, the orientation of bivalence is random, which helps to promote genetic variation as well.
One of 2 or more alternative forms of a gene that arise by mutation and are found at the same place on a chromosome is an allele. The red and blue diagrams represent homologous chromosomes. These chromosomes have the same length, the same centromere position, and the same gene content. However, they have different combinations of alleles.
Firstly, homologous chromosomes pair up with each other. Each chromosome consists of 2 chromatids. This pairing process is called synapsis and is followed by crossing over, which exchanges genes between the chromatids. As the chromatids are not identical, some alleles are likely to be different, which therefore produces chromatids with new combinations of alleles.
The chiasmata is a point at which paired chromosomes remain in contact during the first metaphase of meiosis, and at which crossing over and exchange of genetic material occurs between the strands.
Homologous chromosomes are 2 chromosomes, one of paternal origin, the other of maternal origin, that are identical in appearance and pair during meiosis. In meiosis 1, sister chromatids remain associated with each other. The homologous chromosomes behave in a coordinated fashion in prophase. Homologous chromosomes exchange DNA, leading to genetic recombination. Meiosis 1 is a reduction division in that it reduces the chromosome number by half. The process that result in the creation of genetic variety of gametes are initiated in meiosis 1. The segregation of homologous chromosomes occurs during anaphase 1, resulting in 2 haploid cells, each with only 1 copy of each homologous pair.
The nucleus that undergoes the first division of meiosis is diploid. It has 2 chromosomes of each type. Chromosomes of the same type are known as homologous chromosomes. Each of the 4 nuclei produced by meiosis has just 1 chromosome of each type. They are haploid. Meiosis involves a halving of the chromosome number. It is therefore known as a reduction division. The cells produced by meiosis 1 have 1 chromosome of each type, so the halving of these chromosomes number happens in the first division.
During meiosis, a diploid nucleus divides twice to produce 4 haploid nuclei. The diploid nucleus contains 2 copies of each gene, but the haploid nuclei contain only 1. If 2 copies of one allele of a gene were present, each of the haploid nuclei will receive 1 copy of this allele. For example, if the 2 alleles were dominant, every gamete will receive 1 copy of a dominant allele. If 2 different alleles were present, each haploid nucleus will receive either one of the alleles or the other allele, not both. For example, if the 2 alleles were heterozygous, 50% of the haploid nuclei would receive 1 dominant and 50% would receive 1 recessive. The separation of alleles into different nuclei is called segregation. It breaks up existing combinations of alleles in a parent and allows new combinations to form in the offspring.
A sister chromatid refers to either of the two identical copies of chromatids formed by the replication of a single chromosome, with both copies joined together by a common centromere. In other words, a sister chromatid may also be said as one half of the duplicated chromosome. After meiosis I, the daughter cells enter meiosis II without passing through interphase. Meiosis II is similar to mitosis in that the replicated chromosome is separated into chromatids. Sister chromatids are separated, but they are likely to be non-identical sister chromatids due to the occurrence of crossing over.
While pairs of homologous chromosomes are condensing inside the nucleus of a cell in the early stages of meiosis, spindle microtubules are growing from the poles of the cell. After the nuclear membrane has broken down, these spindle microtubules attach to the centromeres of the chromosomes. The attachment of the spindle microtubules is not the same as in mitosis. The principles are these. Each chromosome is attached to one pole only, not to both. The two homologous chromosomes in a bivalent are attached to different poles. The pole to which each chromosome is attached depends on which way the pair of chromosomes is facing. This is called the orientation. The orientation of bivalence is random, so each chromosome has an equal chance of attaching to each pole, and eventually of being pulled to it. The orientation of one bivalent does not affect other bivalents.
Mendel's law of independent assortment states that genes do not influence each other with regard to the sorting of alleles into gametes. Every possible combination of alleles for every gene is equally likely to occur. When Mendel's work was rediscovered at the start of the 20th century, the mechanism that causes independent assortment of unlinked genes was soon identified. Observations of meiosis in a grasshopper had shown that homologous chromosomes pair up during meiosis and then separate, moving to different poles. The pole to which each chromosome is a pair moves depends on the way the pair is facing. This is random. Also, the direction in which one pair is facing does not affect the direction in which any of the other pairs are facing. This is called independent orientation.
Alright, you ready? In meiosis, one diploid nucleus, in which each chromosome consists of two chromatids, divides twice to produce four haploid nuclei, in which each chromosome consists of one chromatid.
In eukaryotic organisms, sexual reproduction involves the process of fertilization. Fertilization is the union of sex cells, or gametes, usually from two different parents. Fertilization doubles the numbers of chromosomes each time it occurs. It would therefore cause a doubling of chromosomes number every generation. If the number was not also halved at some stage in the life cycle, this halving of the chromosomes number happens during meiosis. Meiosis can happen at any stage during a sexual life cycle, but in animals it happens during the process of creating the gametes. Body cells are therefore diploid and have two copies of most genes. Meiosis is a complex process and it is not at the moment clear how it is developed. What is clear is that the evolution was a crucial step in the origin of eukaryotes. Without meiosis, there could not be a fusion of gametes, and a sexual life cycle of eukaryotes could not occur.
When male and female gametes fuse, their nuclei join together, doubling the chromosome number. The nucleus of the zygote contains two chromosomes of each type, so is diploid. It contains also two alleles of each gene. If there were two alleles of a gene, uppercase and lowercase a, the zygote could contain two copies of either allele or one of each. The three possible combinations are uppercase, 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 lowercase, and lowercase, lowercase. Some genes have more than two alleles. For example, the gene for ABO blood groups in humans has three alleles, I to the A, I to the B, and I. This gives 6 possible combinations of alleles. 3 with 2 of the same allele, I to the A, I to the A, I to the B, I to the B, and I, and 3 with 2 different alleles, I to the A, I to the B, I to the A, I, and I to the B, I.
When two parents have a child, they know that it will inherit an unpredictable mixture of characteristics from each of them. Much of the unpredictability is due to meiosis. Every gamete is produced by a parent and has a new combination of alleles. Meiosis is a source of endless genetic variation. Apart from the genes on the X and Y chromosomes, humans have two copies of each gene. In some cases, the two copies are the same allele, and there will be one copy of the allele in every gamete produced by the parent. There are likely to be thousands of genes in a parent's genome, where the two alleles are different. Each of the two alleles has an equal chance of being passed on in a gamete. Let us suppose that there is a gene with the alleles caps A and undercase A. Half of the gametes produced by the parent will contain A and half will contain lowercase a. Let us now suppose that there will be another gene with the alleles uppercase B and lowercase b. Again, half of the gametes will contain uppercase B and half lowercase b. However, meiosis can result in gametes with different combinations of these genes. A, uppercase B, uppercase A, lowercase a, uppercase B, and lowercase a and b. There are two processes in meiosis that generate this diversity. This genetic variation is pictured here.